Okay, year 13. Hello again. Are you ready for your third biology nugget? This time we're not in the garden because I wanted to show you a little experiment I've been doing during this period of enforced isolation, and that is an experiment I've been doing on myself. Okay, so you may well be aware of lateral flow tests. In fact, that's how I knew that I was positive before I got a PCR. And I wouldn't have known it was COVID unless I had had those lateral flow tests that I was doing standard for a teacher. So good advert for why we should all be doing the lateral flow tests. It's also the case that you might have seen in the news just this morning that there was a study that's come out from one of the London universities that's basically said that actually they're very, very good. They're very accurate at identifying people who are infectious, which I've long said that is absolutely the truth. And people talk about them being this percentage effective, that percentage effective. It's not actually the case. For more on that, go and watch some of the other videos on the channel. They're just testing for different things. I mean, what they're using is they're using immune biology. Basically, they're using antibody testing for the antigen on COVID. So they're testing for the protein spikes directly rather than what PCR does, which is look for the genetic material. But what I decided to do was to carry on doing lateral flow tests for the entire duration of my isolation. Okay, so I've decided to do one test a day because obviously the stronger the band is, the more virus that there is there. I mean, they are what we say semi-quantitative in that way, in that, you know, if you've got more virus there, it's going to stick more antigen, stick more antigen to the antibody and going to stick more antibodies to the test strip. Okay, so you're going to get a darker band, and that is exactly what I've seen. And this is an absolutely fascinating example of a secondary response in biology. So I'm going to show you now exactly what has come up over the last 10 days. Okay, so here they are, all my dated lateral flow tests, as you can see. That was Tuesday the 5th. That was the last routine test I took that was negative, as you can see. And then moving down, we've got Wednesday the 6th, Thursday 7th, Friday the 8th, and obviously all my antigen testing equipment is all over here, right, ready to be used again. Right, so working our way down, as you can see, look, that Wednesday, that was what tipped me off. That is a pretty strong positive. Coming down, Thursday the 7th, here and the 8th, you can see we've still got bands, but they're getting much weaker as we move down. Saturday the 9th, you can just about see a band on that one, but it's very weak. And then you might not see it in this light for Sunday the 10th, but there's no band there. And then Monday the 11th, nothing. Tuesday, Wednesday, nothing. So according to this, I am no longer infectious. So to explain what's happening with all of that, I have drawn you a handy graph. Kapow! Right, everybody loves a graph. So here's my graph. I'll talk you through it. Look, now the 10 days here down at the bottom that you're supposed to isolate for with COVID are kind of arbitrary, but basically that's the time it supposedly takes people to get over COVID. Okay, and that's if they are having what's called a primary immune response. So they're unvaccinated and they're having an immune response that's happening the first time. So they have to make the correct white blood cells. They've got to make the lymphocytes. They've got to make the antibodies. Okay, so what that would look like would be this big red line here with the word primary written on. Now, obviously, this is all very rough and very sketched, okay, because it is semi-quantitative in that I know how many antibodies I've got, roughly by the strength of the band, but it's not, you know, an actual number. So down here, look, we can see I've got where the virus started to replicate, and then this sort of button, this sort of line I've drawn here is an arbitrary line for the threshold of sensitivity beyond which you can actually see a band on the lateral flow test. Okay, so as you can see, in either case, a primary response or a secondary response, the virus would start rapidly replicating and shoot right up here. Now the difference happens up the top here. Okay, now if you are if you've basically got a primary response, right, where you're having to make the antibodies from scratch, there's going to be this lag time here, look, as you can see. So we've got that sort of big flat, hardly any antibodies being produced as the body manufactures the antibodies and the white blood cells from scratch, and then your antibody concentration is going to shoot up and hopefully stay high for long after the virus is gone, and that'll bring down the amount of viruses, right, in your body. And then by sort of 10 days, maybe you'll come down to zero, sort of a bit further back, depending how good your immune system is. But by 10 days, you should be non-infectious. But of course, I, like a great number of people, I'm double vaccinated, so thank you very much, AstraZeneca, for that. So essentially, I already knew how to make the antibodies against COVID. So my lag time, as you can see here on the blue line, right, this is antibodies for a secondary response, is much, much shorter. So antibodies start to get produced here instead, which basically means that I'll still probably get the same shooting up of viral load. It might not reach quite as high, but then almost immediately, because the blue line's coming up really, really fast, you'll start to see the secondary response kick in and bring the viruses down below the threshold of what can be detected by sort of the 5th day there. And to illustrate that why this is such a good demonstration of this, we can actually overlay one on top of the other. So if I take the one from the 5th, where I was negative, that's day 0. So we'll put that down there. Okay. We'll also then take day 1, as you can see. So line that up with day 1 and pop it right the way up there. Okay. So some point between sort of day 1 and day 2, I was at my highest viral load there, as you can see. Now, if we get day 2 and we pop that on there as well, as you can see, it's already started to come down. So around day 2 here, right, the line has started to decline slightly. So I'm going to pop you on there. Day 3, and if you can see that, there is still a line there, but it's not anywhere near as high. So I'm going to pop you there. Day 4, similar thing. You can just about see a line if you squint. So as you can see, look, coming down rapidly. Day 5, well, no line there. So I'm going to pop you right back down, same as that one. So that is a perfect chart. And then for completeness, I can put day 6, day 7 and day 8 in as well. That, for completeness, I would say, is an absolutely textbook secondary immune response. Okay? So there you go. That's a textbook secondary response. I found that quite interesting. I hope you did too. If not, well, who cares? You can go back to your exam questions. But I will see you, certainly, certified non-infectious next Monday. So thanks very much, and I will see you all soon. I am very much looking forward to it at this stage. Cheers. Bye-bye.